not be complete without the Oxford vaccine. Yeah. And so the Oxford vaccine is going to be this weird made up word right over here. There we go. And it stands for, if I remember right, chimpanzee adenovirus, and then the strain is Oxford number 1 or OX1. So adenoviruses are associated with the common cold, but you will already have been exposed to a lot of human adenoviruses, and so I think the idea is that you probably don't already have immunity to a chimpanzee adenovirus. And what they're doing is they're hollowing out the adenovirus. They're using its shell as like a little delivery vehicle, and inside of it, they're putting the gene for the SARS coronavirus 2 spike gene. So there we go. And they're using this to just deliver the spike gene, and so it's just like a different version of what Moderna is doing. They're delivering a messenger RNA to make the spike, and this Chad Oxford vaccine is delivering the DNA to make the RNA to make the spike protein. So same goal, slightly different method. And let's dig in, see what works. All right. So that's the paper. It's freely available. You can go and have a look. And as we do, let's just look at the figures, because they concentrate all of the useful data. Well, not all the useful data. Yeah, there's always little tiny devils in the details. Anyway, this is from the inoculation. So they're putting lots of these little adenoviruses in. These things are not able to grow, if I remember correctly. They are just delivery vehicles. So it's a little protein-coated capsule that delivers a gene into a cell. That is the idea, and they're very good at their job, and probably people would not have pre-existing antibodies to many chimpanzee adenoviruses, but it looks like they can get into human cells anyway, so great. What are the main symptoms, or the main side effects? Well, it looks like pain is one of them, and it looks like tenderness. Oh, yeah, need a little tenderness. It looks like pretty much everybody had a sore arm after they injected this. Assuming they injected it in the arm, I don't know. That's one of the details you can go and dig up. And here is another set. And so this is different adverse reactions in the first 7 days after vaccination that the patients reported themselves. And so patients are also saying that they had some chills and fatigue and fever and headaches, which are common with different kinds of vaccines. You could make a generic immune response, and it would provoke a lot of this stuff. Interesting thing is that fever's fairly low. Now, they had these people either just reporting the symptoms as is, or they had them taking paracetamol, which I think is acetaminophen, and also reporting results. There's maybe a little difference in severity, but I don't think there is a giant difference between the two groups. More, they're going into specific symptoms. In general, they look perhaps similar, but maybe not quite as bad. But the neat thing is, so over here on the first dose, you get a lot of symptoms. But then by the time they get to the second dose, look at that, lower symptoms. So that is interesting. All right. And here we go. Actual results. That's what you came here for, isn't it? So we're starting out, and so these are samples where they were not given any virus. And then they're counting the amount of antibodies. And so you can see right up here, we're getting up to about a level of 1,000. Remember, 1,000 is low, and we got up to levels of 1,000,000, but each test is a little bit different. Everybody's using different versions of the test, and so you can have different numbers for the same serum, theoretically, in two different people's hands. I think this is probably going to be internally consistent, so all the numbers within here will make sense relative to each other, but you can't necessarily compare all the numbers from one test to another, or you've got to keep that grain of salt in your pocket while you're doing it. They do a thing here which is maybe, I don't know, a step more open and fair than Moderna. So they're still biasing the sample pool. They've got a lot of people in here that they state right up front. You don't have to dig in the supplementary appendix, but they state that they were asymptomatic. But they also mark out where the severe cases are. So it's these little blue squares that you can see are all here, mostly above the average levels that you get with either one vaccination or two vaccinations. It looks like a second vaccination does give you a higher response. You get up to around 1,000 as opposed to around 100 with the first vaccination. So yeah, that's a nice thing. And the side effects weren't as bad in the second vaccination. So maybe this is a thing that you can just get vaccinated with. I don't know, more than once anyway, maybe 2 or 3 times, and get better immunity. But I think overall, the total antibody numbers are a little bit underwhelming. They are lower, considerably lower than a person who's had severe COVID. And I don't believe we know exactly how many days it has been since these people had COVID. And so we don't know if these are kind of their peak amount of antibodies or if this is what's left after, a couple of months, something like that. Okay, zip into the next part, and we'll jump past this one to this one because it's looking at similar information, and it sums it all up very nicely. So this is neutralization. Remember I said we want to be somewhere between like 500 and 1,000 in terms of neutralization. On this one, they have a couple of severe cases here. So they got 3 severe cases that are up about where we would expect numbers at or north of 1,000 for the neutralization titer. They got one with severe symptoms that seems to have no neutralizing antibody, which is kind of neat. Haven't seen one of those before, but yeah, look long enough and you will find every possible permutation. So they're showing that on average it looks like this virus is giving about the same numbers, which look like, I don't know, 250, 300, something like that, similar to the Moderna numbers. But once again, most of the people in this green scatter up here are people that had mild or even asymptomatic COVID and who probably have a very weak response and who are probably susceptible to reinfection. Or much more likely to get reinfection sooner than the people with the blue dots up here, based just on antibody levels and T-cell levels that we know about. So once again, kind of underwhelming, but positive. They're getting a response, and yeah, you got to like something about that. I think the other thing to point out is that here, this one shows it very nicely. The neutralization numbers are dropping off a little bit over time, as were look at the total antibody numbers. So right after the second vaccination, they're pretty good. A couple weeks after that, and then they start going down, and these numbers are also going down. It's the same old story, and so it's kind of a case of you want to try to get to probably the highest level you can get to, and then you know that this number is going to decay fairly rapidly over time. And you just have to deal with that, and when it gets low enough, you would have to revaccinate. So it's probably, these both look like maybe once a year kind of vaccinations. Let's see, the numbers over here are actually for T cells, and they're counting them, I think, per million, if that's right. There we go. Spot forming cells per million. Normally people would do this in terms of percent, and so 1 out of 100 is what? 10 million out of a million? No, 1 million out of 100 million. Yeah, 10 million. 10,000. There we go. 10,000 out of a million. That sounds right. Yeah, that's the equivalent. And so that's where we would want to be in terms of T cells. And look, this person, for one brief shining moment, 2 weeks after the first inoculation, but before the second inoculation, had really good T cell numbers. For everybody else, they're down not at like 1%, which is, I think, about where you are normally right after coming out of an actual infection that's moderate or severe. Here's the 0.1% level, and you've got some people that are at that level, and for the most part, we're down closer to between 1/100% and 0.1%, which is, I've got to say, not much of a T cell response. So yeah, once again, it's kind of a one-sided response. The white blood cells are not great. The antibodies are better, but maybe not as high as we would want them to be. But they're getting some effect, and so this vaccine, as with the other one, may protect against like severe symptoms. Maybe that's where the bar is at the moment. I don't know that this would definitely protect against infection although some of the people at the higher end may indeed be protected. It's just a question of how long that window is when they would be protected. So there you go. That is the chimpanzee adenovirus OX1 vaccine against SARS coronavirus 2. And we put these both out on the same day, the Moderna and this one. And you can compare them. There was a third vaccine that was reported. It was an adenovirus 5 vaccine, very similar to this, from a Chinese company. I don't know if their protocol was a little different, but their results I thought were maybe a peg below what you're seeing in this particular paper. So it's at least some hope, right? And the world needs hope. So thanks very much. This has been Ask Dr. Ben, and this was an Ask Dr. Ben update.